Good morning, students. Now we have to move on to the next class of algae, that is Bacillariophyceae. So under Bacillariophyceae, we have to study one type specimen, and that is Pinnularia. First, let us see the systematic position of Pinnularia. Pinnularia, the algae, it comes under the division Bacillariophyta, and the class is Bacillariophyceae. Order Pinnales, family Naviculoideae, and genus is Pinnularia. Pinnularia, the algae, it is a unicellular algae, and most of the species coming under this genus are of freshwater forms. This is how the cell of Pinnularia looks like. As you can see here, the cells are much longer than broader. So they are much elongated type of cells, and the cell wall of these Pinnularia or the members of Bacillariophyceae, they are impregnated with silica, and the cell wall of these are called frustule. Cell wall is called frustule and it is impregnated with silica. And this cell wall, it consists of two valves. Two valves are there, like you can see here. Two valves are there for the frustule, and one valve is slightly larger than the other. As you can see here, it fits the larger one, and the smaller one are seen here. Smaller one can fit into the larger one, and it resembles the arrangement. This arrangement resembles like that of a box, a lid and a box with its lid, right? And the upper valve is called epitheca and lower valve is called hypotheca. And each valve also has two parts, namely a flat surface called the valve face and its margin called mantle or girdle. Okay, this is the margin, this is called girdle, and this face is called valve face. Okay, and we can see some lines here, right? And these are called striations, or these striations are called, or these markings are called striae. These are nothing but parallelly arranged ribs or transversely parallelly arranged ribs, and these are called striae. It extends all along the length of the Pinnularia. Each of the rib consists of a row of very small pores. Each of these striae, or each of these parallel lines, it consists of small pores, and that pores are called as punctae. And there is a longitudinal line. You can see here, there is a longitudinal line here extending along the length of the algae, and that longitudinal line is called raphe. And raphe has wider outer and inner ends and narrow middle portion. Okay, that is clearly demonstrated here. It has three rounded wall thickenings or swelling, right? These are called nodules. These swellings are called nodules. One of them is situated at the center or the midpoint. See here, this raphe has three swellings or three nodules. The center one, or the nodule which is found at the central portion of the thallus, it is called as central nodule, and those which are found at the two extreme ends, they are called as polar nodules. It helps in this raphe, it helps in the gliding movement, or this helps the algae to glide along the surface. As we have already mentioned, the cell wall, it is silicide. that is silica depositions are found, and so it is very rough in its nature. And inside the cell wall it has protoplast, and the cytoplasm is along the periphery, and in the central portion it has a long or large vacuole. Two large and elongated chloroplasts are also there, which are green or brown in color. Pyrenoids are also found in the protoplasm. That is all about the structure of cell. Okay. Now we have to move on to the reproduction. Reproduction here occurs by way of fission, that is vegetative reproduction is taking place here through mitotic division. But it is a special case here. It occurs usually at the midnight. During cell division, what happens is the cell increases in size, that is at right angle to the girdle. Cell increases its size at right angle to the girdle. And nucleus mitotically divided and two daughter nuclei are formed, and the two valves of the cell, they move apart. As you can see here, this is the parent cell, this is the hypotheca, and this is the epitheca. Inside the cell it undergoes mitosis and the nucleus divides and forms two daughter nuclei, and what happens is it elongates. Firstly it elongates along at right angle to the girdle, and then mitotic division occurs, two nuclei are formed, and then these two valves, they separate from each other, and the protoplasm is divided in such a way that each of the valve, they get half of the protoplasm. So protoplasm divides into two bits, and one bit goes to one valve and the other moves on to the other valve. So each of the valve receives one portion of the protoplasm. And now we have two daughter cells which are having one of the valve and the one nucleus with protoplasm, one nucleus with a bit of cytoplasm. Likewise here also we have one valve with nucleus and a bit of cytoplasm. So one end of both the daughter nuclei will be naked now, right? Only one valve will be present now. So they have to synthesize. The new valve has to be synthesized. The newly synthesized valve will always become the hypotheca of the daughter cell. The daughter cell which receives the epitheca of the parent will become as large as the parent cell. And on the other hand, the other cell which receives the hypotheca of the parent cell will be much smaller than the parent cell. Right? Here you can see two valves. One of this valve goes to this daughter cell and the other goes to this daughter cell. And how in whichever case it may be, the two daughter cells are having only one valve at this point. Now they have to synthesize the other valve. So the newly synthesized valve, whether it may be in this case or whether it may be in this case, the newly synthesized valve will always become the hypotheca. Okay, here this is the newly synthesized valve, and here this is the newly synthesized valve. Okay, so whichever may be the case or whichever may be the position, always the newly synthesized valve will become the hypotheca. So the daughter cell which have received the epitheca of the parent cell, this is the epitheca. The daughter cell which has received the epitheca of the parent cell will, as generation pass on, it will become smaller and smaller, right? So always the cell which receives the epitheca of the parent cell will be much smaller than the parent cell, and thus continued cell division in this case, it creates a line of daughter cells which will be progressively decreasing in its size, and after a point it will reach a limit whereon further decrease in size cannot be possible. At that case, what happens is they go on for another mechanism, that is oxospore formation. Thus, oxospore formations are often called the renewal spores or rejuvenescent cells. They are formed only when the individual's cells they get diminished to its minimum size. Maximum minimum size means to the limit beyond which it cannot decrease in its size. At that point, they go for oxospore formation. And during oxospore formation, each of the cell they secrete certain amount of mucilage, and the successive mucilage production continues. It goes on, and the production causes two valves of the cells to split apart. They separate, and the protoplasm it becomes naked. This protoplast now increases to its maximum size, and later on becomes surrounded by a silica deposited pectic membrane or a silicified pectic membrane, and this membrane is called perizonium. The nucleus does not divide, and the protoplasm secretes fresh epitheca and hypotheca, and thus a larger new individual is formed. No sexual reproduction is found in Pinnularia. So only method of reproduction is vegetative reproduction, that also through fission. And there is a specialty in fission that we have gone through now. It has two valves. It divides, protoplasm divides mitotically, and two nuclei formed. One of the nuclei and a bit of cytoplasm is given to each of the daughter cells. But now at this point, the protoplast or the daughter cells are having only one wall. The other side is naked, and new valve has to be secreted or synthesized, and the newly formed valve will always become the hypotheca of the daughter cell. And in this way, two of the two daughter cells, one will be smaller when compared to the parent cell, and one will be larger compared to the parent cell. So in this way, one cell line or the one line of the daughter cells will become progressively smaller and smaller. Thus there will be a progressive decrease in size of one cell line, and at certain point there comes a situation like that the cells they cannot diminish further, or they cannot decrease in their size further. So at that point they go on for the production of oxospores. Oxospores are also known as renewal spores or rejuvenescent cells. Okay, this is all about Pinnularia. Thank you.